Intercellular communication can be defined as conversation between two cells. It is studied under the branch of cell biology, which encompasses the study of cellular organelles and cell signaling. Intercellular as well as intracellular communication both come under cell signaling. Intracellular communication can be defined as the communication that takes place within the cell. For example, the cellular response that occurs in response to molecules present inside the cell. Cells communicate through chemical signals. These chemical signals are known as ligands. These ligands can only interact with the cell that has the receptor for the particular ligand. This ligand receptor association provides the specificity to the cell signaling or intracellular communication. There are following types of intercellular communication that occurs between the cell: paracrine, autocrine, endocrine, and cell-to-cell contact signaling. Features of cell signaling. According to the intercellular communication definition, there are following characteristic features that define intercellular communication. That is specificity. It is attributed to the high affinity of the signaling molecule, that is ligand, and the complementary receptor. The complementarity of the structure between the ligand and the receptor binding contributes to the high specificity of the signaling pathway. Another character is amplification. During intracellular communication, the signal amplifies several folds to generate the response. Amplification can be attributed to the cooperative nature of the receptor-ligand association. There can be a desensitization, also known as adaptation. It is the condition that arises due to the continuous binding of the ligand to the receptor. It can result in lack of response by the cell. And to avoid such condition, cells follow feedback regulation, which allows the removal of ligand and receptor when needed. Another character can be integration. It can be defined as the ability of the system to receive multiple signals and produce unified response appropriate to the needs of the cell. Then there are different stages of intercellular communication. There are three main stages of communication. They are as follows. First one is reception. It refers to the receiving the signal via ligand molecule binding to the receptor. Receptor proteins are the molecules that spans through the plasma membrane. This receptor provides a specific binding site for ligand. The ligand generally undergoes conformational change, leading to the sequential activation of protein cascade. Another common method is phosphorylation and dephosphorylation of the intracellular proteins. Then there will be transduction. It refers to the transfer of signals from the cell surface to the interior of the cell, and it is achieved by activating several proteins via phosphorylation and dephosphorylation. A common method is the production of the secondary messenger. This ensures the amplification of the signal received. Then there is a response. A cell generates a varying type of response according to the need. It includes transcription and translation of the protein, or inhibit the synthesis of certain protein, secession, or inhibition of cell cycle. The result is the change in the metabolic activity of the cell. So these are the different phases of first one is reception, where the ligand binds to a receptor. So then there will be transduction, where the conformational changes in the receptor, they are going to activate the cascade of protein kinase synthesis, which are usually the secondary messengers. They give rise to active effector, for example, transcription factor. So that is going to act on the genes which will be responsible for production of the specific proteins as a response. And this is the response phase. The examples of signals to which cell respond. There are the following examples of the ligand to which cell respond. So these are antigens. They are going to respond to antigen which leads to activation of the, for example, uh, CD4 cells, cell surface glycoproteins, the developmental signals, growth factors, hormones, neurotransmitters, pheromones, nutrients, mechanical touch, or light. They can act as the stimulus. Example of intracellular communication. 6 basic receptor types can be used as example of intercellular communication, and they are as follows. The G protein coupled receptor used in vision transmission, epinephrine metabolism. These are the examples. Then receptors tyrosine kinase used in the cell division and glucose metabolism. And then receptor guanylyl cyclase used in the metabolism of nitric oxide and RO system. Then gated ion channels, which are used in the transmission of the nerve impulse. Then adhesion receptors used to maintain the integrity and the development of embryonic stages. Then nuclear receptors, which are usually the steroid receptors, used inactivation and inhibition transcription of certain proteins. So these are the examples of the mechanisms. So this is example of a ligand gated ion channel. Okay, so ultimately the ligand, specific ligand, binds to the receptor, and by transporting this particular ion, it gives the response. So then the G protein coupled receptors are the transmembrane proteins. Then by activating on the secondary messengers, it gives its response. Then the nuclear hormone receptors, which the ligand can cross cell membrane and they can bind to directly the receptor in the nucleus. So then we have receptor tyrokinase, tyrosine kinases, okay, so which are going to bind to the specific hormones, and then by activating certain second messengers, they respond. So this is another example of the G protein receptor activation. Okay, so they can have a multiple modality of giving a response. By it may increase the cyclic AMP levels, it may increase DAG levels, or it can increase the Rho levels. Then the response can be, there can be a variant, the protein channels or ion channels, okay, so which are, they can be ligand gated, they can be voltage gated, they can respond at a specific voltage, and they are going to allow only the specific ions to pass through. Then some of the ion channels may be mechanically gated. When pressure increases, these channels, they are going to open, and sometimes the channels may be all the time open for specific ligands. And also there are the cell adhesion molecules, so which also going to help in the connectivity. So these are carins, selectins, Ig superfamily, and integrin. Okay, so they are going to help in mainly the integrity of the cell. Types of intercellular communication. The intercellular communication types can be categorized into 3 types as described: autocrine, paracrine, endocrine signaling, and also cell-to-cell contact signaling. This signaling mechanism is used according to the ligand and need of the cell. Paracrine signaling. It is the signaling pathway that has ligand molecules traveling a short distance to bind to its receptor. It can be defined as a signaling pathway in which cells communicate over a relatively short distance. The important feature of this signaling pathway is that it allows the cell to be in coordination with other cells neighboring it. One of the most important roles of paracrine signaling is in embryonic development. It is through the pluripotent cell that decides its cell lineage, which it follows to develop specific organs and tissues. Another most widely studied example is synaptic signaling, which comes under as a type of paracrine signaling. Synaptic signaling is the method used by nerves to transmit their impulse or action potential across nerve to another. The name is derived from the synapse. Synapses are the junction between two nerves. It contains a neurotransmitter that acts as a ligand, which then binds to the receptor present on the membrane of the second nerve, that is postsynaptic membrane. The common ligand that is used is acetylcholine, and the gated ion receptor mediates the signal transduction. The binding of a neurotransmitter opens the gated ion channel, which changes the electrical potential of cell, and thus the signal travels from one nerve to another nerve. Autocrine signaling. Autocrine signaling is a type of signaling where the cell releases a chemical molecule which acts on the ligand that binds to the receptor on the cell that produces it. This type of signaling is generally observed in the self-activatory molecule and as a part of immune response. The most important example of autocrine signaling is apoptosis. It can be defined as programmed cell death. In this case, phosphatidylserine, a molecule present on the inner leaflet of plasma membrane, moves towards the extracellular side, acting as a ligand for apoptosis. It also plays an important role in the immune response. Cytokine production and the regulation is mediated by the autocrine signaling pathway. Then endocrine signaling. It's a long distance signaling pathway, and the ligand molecules is transported over a long distance to which it binds to the receptor. The best example of this type of signaling is hormones. Hormones from various glands are poured into the bloodstream, and then they travel along with it. When they reach the target cell, they bind to the receptors, generating appropriate response. Then cell-to-cell contact signaling. It occurs when two cells are connected through gap junctions or plasmadesmata in case of plants. These junctions provide a channel through which ligand molecules can travel. And ligand in this case is known as intercellular mediators, which brings the response. So these are the example of different modality of intercellular connection. As we discussed, it can be autocrine, it can be through the gap junction, it can be a paracrine modality, it can be endocrine. So autocrine, a cell target itself, the cytokines or chemical mediators produced by a cell, they will have a receptor for the same cytokine that is going to bring the cellular changes. And the two cells, adjacent cells, sometimes they can communicate through the gap junction, and sometimes the action potential or nutrient materials can be transmitted through these particular gap junctions. So then paracrine, where the chemical mediators are produced by a particular cell, they're going to affect the functioning of the adjacent cell to which they will have a receptors, and this is called as paracrine functioning. Whereas endocrine functioning, where the chemical substance produced from particular cell, they are poured into the bloodstream, and they're going to reach the far away target cell and bind to the receptor, and this receptor complex brings the changes in the distant cell. This is another example of paracrine function, where which is called as synapse, one nerve fiber, and it is going to activate the adjacent nerve fibers through the synaptic cleft, where the part of the nerve is going to secrete neurotransmitter, and this neurotransmitter is going to bind to the postsynaptic receptors, and they brings the changes in the postsynaptic membrane.